Hello everyone. In this video, we'll talk about bioequivalence. So this is actually an introduction class for the topic, and we'll go through two main concepts, which is about the overview of bioequivalence, which is BE, and also about the pharmaceutical factors that can influence BE. I am Dr. Yuen, and welcome to my channel of Lighthouse Pharmacology. Why do we need BE studies? So historically, it's found that identical tablets and capsules products, once manufactured by different manufacturer, i.e. it's at different factories, they are found to have significant differences in their dissolution rates for the active ingredients. These are found for the important drugs such as digoxin, tolbutamide, chloramphenicol, and tetracycline. Therefore, we start to have some legal requirements on the registration of the generic drugs to ensure that they are safe to be used. When a generic drug is claimed to be bioequivalent to a reference drug, it is assumed that they are therapeutically equivalent. Therefore, gives confidence to the doctor or the consumers that once you change your medication, if it's the same medication from the reference originator into a cheaper generic product, it should provide the same therapeutic effects. Let's go through some basic concepts. The first one is about pharmaceutical equivalence, meaning that for both things, there's actually equal amount of identical active drug ingredient, the same salt or ester. They are also provided in the identical dosage form as well. In the concept of BE, we mainly carry out studies on the oral tablet dosage form, for example. But they may not contain the same inactive ingredients or what we call excipients. When we compare the reference drug on the left and the test drug, which has not been granted as generic yet because it's still under testing, the possible differences include the drug particle size, the excipients, the manufacturing process, the process of actually making it, and also the site of manufacturing it. All these factors can actually collectively contribute to the different performance in vivo. Another concept is about pharmaceutical alternatives. When sometimes they have the same therapeutic moiety or precursor, but they may not have the same, be in the same ester or salt form, or the amount, or even the dosage form. When we look at this summary diagram, we can see that the first one is on the pharmaceutical equivalent, where most of the things are equal. Therefore, that can contribute to an bioequivalent product. Similarly, for the pharmaceutical alternative as well, we can imagine there's probably more tests that you need to do in the formulation site to ensure that when they do a bioequivalent study on the pharmaceutical alternative, you actually get the same results. There's another concept here on therapeutic equivalent. So this is actually, it's more like already a clinical trial study because you actually have to measure the same clinical effect. So it's not just about the concentration of the drug after it's being administered or it's being taken, but it's also about the same clinical effects that needs to be done. So this, as you can expect in a way, requires more cost because it has to be a proper clinical trial-like setting. So this is something that most of the generic companies will try to avoid actually because of the cost involved. Going back to BE, so this is the general definition of it. When there's two medicinal products which are bioequivalent, so meaning you have to make sure that their bioavailability is similar as well, so they can contribute the same efficacy and also the safetiness as well in terms of the drug. Okay, so when do you need to do BE studies? So in the first scenario here, it's during the development of a new active substance. So BE studies is more like a bridging study, so to make sure that in the clinical formulation of it will be similar as well for the product that you're trying to get. The second scenario is the more common one, whereby we look at the comparison of a generic drug in a way with an approved active substance, so whether the formulation is an immediate release or the modified release forms. So next we look at the reference product itself. So the reference one is like the originator product who developed the drug, so it's protected by patent and It can be marketed under a brand name. So in order for this original product to be approved, they actually have to submit a lot of clinical efficacy and safety profiles as well. And they have to do the standard clinical trials of phase 1, phase 2, and phase 3. So you can see most of them, it's actually by the big brand names that we know, like Pfizer, AstraZeneca, MSD, and so on. Next, we'll look at the generic drugs, which are mainly manufactured by some local players like Pharmaniaga or some overseas players like Ranbaxy as well. The drug products, have shown to be bioequivalent. That's how they can be registered in the generic drug list. They have to be equivalent to the reference drug in their active ingredient, route of administration, dosage form, their strength, their indication, and their safety profile. They may have different excipients or inactive ingredients, different color and shapes. And we take note that there's almost half of the marketed drugs are actually generics. Right. So what are the main differences between the reference and generic drugs? So the main thing is about the pricing actually. So because there's a lot of R&D which has been invested to be able to develop the reference drug. So obviously the costing, the company needs to recover the cost as well to ensure the development of newer drugs as well. So the costing of it is expensive. But the generic drug is more like a photocopier in a way concept, so it's cheaper. Only the cost of manufacturing is required. However, for reference drugs, we have to understand that during the drug development process, they could be screening thousands of new drug candidates, but eventually it's slowly being streamlined and reduced until the one best balance in the efficacy and safety profile drug can be move on to the approval and be used in the market as well. So therefore, there's a lot of costing involved because it takes years for that. So it's protected by a drug patent, right? Then after the patent is expired, so any drug company can manufacture and formulate their own version of the same drug, but with different excipients and all, and do a BE study to get it registered and approved in the market. In summary, BE studies is a must to be done to ensure that the clinical and the therapeutic effects of the generic drugs are comparable to the originator or the innovator drugs. So the therapeutic effects is directly reflected by the similarity in their drug plasma concentration profile. So the drug plasma concentration depends on the bioavailability of the product, whereby the bioavailability of the drug in the body depends on the formulation itself, which is reflected by the pharmacokinetic process. The overall assumption is that if they can produce a similar plasma concentration drug graph, the similar concentration of the drug can actually reach the site of action to produce the intended therapeutic effects. The main advantage of the generic drugs is because of its costing, so therefore it's more affordable for a greater population of patients. When we look at the graphs, so this is one of the example. Basically, the drug is given to most of the time healthy volunteers, right? There's two different groups and they'll be given two different formulations and there'll be a crossover. Eventually, the same person will actually get the two different samples of the drug at different times, and the drug concentration profile in the blood will be tested and the graph will be drawn. Parameters such as the area under the curve, which is AUC, and C max, the maximum concentration, T max, the time to reach the maximum concentration. These parameters are noted and there's a certain range that they are being accepted for their similarity. Further information and details on the design of the BE study and also the analysis of the graphs and data will be covered in subsequent classes. Let's move on about the bioavailability. As we know, the bioavailability is the direct influencer of the bioequivalence. So there are other factors which can contribute to the differences in the bioavailability, which is BA, that includes the particle size, manufacturing process steps, the excipients, and also the crystal or salt forms. So based on the definition by FDA, so BA is defined as the rate and extent of the active ingredient or the active moiety is absorbed, right? and it's available at the site of action. Over here in the equation, BA, which is reflected by F, it's defined as the area under the curve of the oral form divided by the IV form. So at the IV form, we assume that the amount of drug absorbed is 100%, because when we look at the chart on top, we can see that there's losses of drug before it can reach the site of action. So for example, in the first stage when the drug is in the stomach, there's some percentage of undissolved drug, and there's also some percentage of non-absorbed drug as well. And also, last but not least, some drugs which are lost during the first pass metabolism in the liver before it can reach the systemic circulation. In the table, we can see that there's lots of factors that can influence the drug bioavailability. So for the topic on BE, we'll only focus on those pharmaceutical factors which we can change in the formulation, but not on those factors that we can't change, like the physiological factors in the body or the drug characteristics itself, like the drug pKa. Let's move on to the second part of the whole video, where we focus on the physiochemical properties of drug particles. So there's a concept that we need to know, that the drug dissolution rate is the main rate limiting step of the drug to reach its site of target. So as you can see here in the flow chart, so dissolution is the process that can occur in any stage of after the drug has been swallowed, whereby the tablet undergoes disintegration and deaggregation for more dissolution to occur. When you translate all this into the graph, it forms a S-shaped curve, whereby the dissolution phase contributes to most of the percentage of dissolved drug, which is the concentration of the drug in the blood. There's some equation, for example, the Noyes-Whitney equation calculates the rate of diffusion of solute through the diffusion layer of a spherical particle. This is the equation. You can see the dm/dt refers to the rate of dissolution, modulated by other factors like A refers to the effective surface area, H the thickness of the diffusion layer, and C for the concentrations. You can see this is where all the parts fit in if you put it into a diagram. We won't be doing calculations of the equation for this module, but we need to know some of the take-home message from here, whereby there are factors like the boundary layer, the solubility in the diffusion layer, the effective surface area, the wettability, the particle size, and the molecular size could contribute to the rate of dissolution. But we have to take note that every drug is different, so they all have different characteristics and patterns. The different solutions is actually different for each individual drug. All of the changes that we need to make in the formulation needs to be tailor-made. Let's look at the first one, which refers to the particle size and surface area. Looking at a typical drug example, in this case, which is called phenacetin, when you look at the graph, the bigger size one at the bottom, you can see that the amount dissolved is actually lesser and slower across the time. Therefore, it will be good to increase the effective surface area with methods such as micronisation to enhance the drug solubility. However, we also need to note that the increase in drug solubility, which refers to an increase in bioavailability, could also increase the incidence of side effects because of the concentration of the drug in the blood has increased. Therefore, in the formulary such as the British Pharmaceutical and all, they actually specify the size range that is permitted for the particular drug. There are some exception scenarios. So for example, in the first one, hydrophobic drugs in medium with poor wetting properties, the reduction of particle size can actually encourage aggregation. When the particles aggregate, reduce the effective surface area, therefore reduce the dissolution rate. So this can be observed in examples such as aspirin, phenacetin, and phenobarbital. How do we solve it? So one of the ways is through micronisation or milling of the drug with a wetting agent or hydrophilic carrier. Therefore, we can actually produce sizes at the nano range. One classical example is on a drug called danazole, whereby the relative bioavailability increased by 400% for a micrometer reduction size to nanometer range. Then there's also other commercial products which uses the same analogy too. Second solution for aggregation of particles is to add a wetting agent into the formulation, such as polysorbate 80, to increase the solvent penetration into the particles, resulting in a larger effective surface area. However, it's worthy to note that the effect of wetting agents are highly drug-specific, but is still routinely added to many formulations. In the second scenario, there are some exceptions whereby for certain drugs, the rate-limiting step is not the dissolution process. Therefore, an increase in the effective surface area of the drug would not further cause an increase in the absorption rate. This is applicable for drugs such as penicillin G and erythromycin, whereby these drugs are unstable in the gastric fluid. When the size is too small, they can actually undergo chemical degradation, which therefore decrease the amount of intact drug available for absorption. There are other general solutions as well. For example, you could form a solid solution or molecular dispersion, whereby the molecules of the sparingly soluble drug can be dispersed into water-soluble drug or to be replaced in its crystal lattice. Therefore, it's a mixture of water plus drug molecule into a structure, or we can actually just simply disperse the drug into a soluble carrier such as PVP. Next is about the factor on particle shape and density. Again, back to the concept of effective surface area, so we'll try to modify the shear rate of the solvent that comes in contact with the solid. It is assumed that the drug solubility is independent of the particle size, and it's governed by this equation called Ostwald-Freundlich equation. On the right-hand side of the simplified version of the equation, as you can see, S refers to solubility of the microparticles. However, if you want to reduce the S, you actually need to reduce the R, which is the radius of the particle, to a micro level before it can actually cause a change in the solubility. Next in line is about the factor on salts. Generally, salts increase the dissolution. The most common form of salts are sodium salts of acidic drugs and hydrochloride salts of basic drugs, where they produce fast and predictable effects. Other salts such as sulfate salts can also be applied. However, there are some salts that actually reduce the solubility, such as the aluminium salts of weak acids and palmoate salts of weak bases. Therefore, it can be used in different scenarios to form tablet coating for sustained release of drugs due to the delayed absorption. However, when choosing the salts, you actually have to consider their chemical stability, hygroscopicity, manufacturability, and crystallinicity, because different salts have different properties of those. For example, the sodium salt of aspirin, called sodium acetylsalicylate, is more prone to hydrolysis than aspirin itself, so therefore it may not be the most suitable salt to be used. Another way to form salts with high dissolution rate and bioavailability is to formulate in situ, or basically to add a basic excipient to the formulation of a weak acid drug, or to add an acidic excipient to weakly basic drugs. For example, addition of basic ingredients such as aluminium dihydroxyamino acetate and magnesium carbonate to aspirin tablets could form a relatively basic diffusion layer around the dissolving particles to allow it to dissolve faster. The last physiochemical character of the drug that we're going to discuss is on the crystal form. The first one is on polymorphism, whereby many drugs can exist in more than one crystalline form. For example, metastable polymorph usually has higher dissolution rate and higher bioavailability than the corresponding stable polymorph. So this can be seen for drugs such as chloramphenicol. On the right hand side here, you can see that the different structure structures or the way that the drug molecules can be arranged into their crystal lattice forms different polymorphs. Second type of crystals are called amorphous solid. The amorphous form usually dissolves more rapidly with higher bioavailability than their crystalline form. As you can see, their structure underneath here, whereby the amorphous solid one is not so nicely or neatly arranged, which also contributes to their instability of structure, as seen for certain classical examples such as novobiosine, whereby the amorphous form could slowly convert itself to the more thermodynamically stable crystalline form, causing with a loss of the therapeutic effects. One way to stabilize the drug in amorphous form is to add polymers such as copovidone into lopinavir and ritonavir. Again, like any other factors, there's always some abnormalities. For example, for the drug of erythromycin acetolate, the amorphous form actually has a lower dissolution rate than the crystalline form, as shown in the graph. The last crystal form that we're going to discuss is on solvates. Basically, it's the different solvents that forms different drug crystals to cause difference in the dissolution rates. If the solvent is water, it can form hydrates. Depends on the number of water molecules, it can form monohydrate or dihydrate. Let's move on to the second factor that can influence the bioavailability or bioequivalence of a drug, which is the manufacturing process or steps. For the method of granulation, wet granulation is commonly used to improve the dissolution rates of poorly soluble drugs. It can add hydrophilic properties to the surface of the granules, and this is considered a better way than the dry or double compression procedure. In the wet granulation process, we can also add other active ingredients such as fillers, diluents that can increase the hydrophilicity to increase the dissolution rate. In the graph on the right hand side, we can see that through different manufacturing processes such as direct compression with sprayed dry lactose or wet granulation or other different excipients that you add on can actually produce different dissolution profile. So based on what you need, so you can actually choose which is the best that suits your need. With other newer tableting machines and materials, there are other factors which are more important than the wet granulation method, such as the critical formulation or the proper mixing sequence of the ingredients or the time to add them actually plays a more important role. The second factor in the manufacturing process is on the compression force. Force applied can influence the apparent density, porosity, hardness, disintegration time, and also the average primary particle size of the compressed tablets. So it's actually a competing relationship between the enhancing effect, whereby you increase in the surface area through the crushing effect, and also the inhibition effect, whereby you increase the particle bonding that increase its density and hardness. As you can see in the different graphs below here, the increase in pressure can cause a decrease in the dissolution rate in general, but there's also some that can actually increase the dissolution rate, and you can see the graph, it can be in a wavy form or a U-shaped form. So it depends really on the formulation that you're playing with. In general, during a high compression force, which is normally accompanied with high pressure and temperature, could form a firmer and more effective sealing layer by the lubricant. Therefore, it actually reduces the solvent permeability to reduce the dissolution rate. In the graph at the lowest line, you can see actually represents the highest force applied during the tableting process caused a reduction in the amount dissolved over time. The last factor that we're looking at is on the different excipients that you can use in your formulation. It's probably one of the most important factor for generic drugs formulation, because that's one of the main things that and ingredients that you can play around with on what to add in or the ratio that you can add in. Excipient refers to all the other ingredients besides the active drug that you add in. Right, so the first one that we're going to talk about is on glidants. So therefore, as the name suggests, helps the tablet materials to glide and flow through uniformly into the dye cavity of the tablet compressor. So hydrophilic glidant materials act better on hydrophilic granules and vice versa for the lipophilic ones. Examples of glidants include cornstarch, silica derivatives, and colloid. Next, we'll talk about fillers. As we know, the active ingredient of the drug is actually present in the tablet at a very small amount, can be up to like 1 mg, 5 mg, which is very, very tiny and impossible to hold. So the fillers act as a bulking agent into the final tablet weight of 100 to about 500 mg. The binding of the drug to the fillers may occur as well that can affect the bioavailability. When you look at the graph on the right hand side, the different percentage of starch actually could increase the amount dissolved as well. The examples of fillers include lactose, sucrose, and also plant cellulose. For binders, it actually provides cohesiveness to the tablet. When you add too little of it, it can form a weak tablet that cannot maintain its integrity, so it can easily be crushed during the transportation process. If you add too much of the binders, it's too strong a tablet in a way that it reduces the release and the dissolution rate. So examples of binders include microcrystalline cellulose, starch, and lactose. The effect of the different binders can be shown in this graph, which they tested on phenobarbital. Depends on which profile that you're looking at. If you want something which can dissolve very, very fast for phenobarbital, the better option would be gelatin. Another class of excipients called disintegrants are often added to the tablets as well to improve the disintegration process when the tablet is exposed to an aqueous environment in the stomach or in the GI tract of small intestines. So the mechanisms that it is used to work is through the amplification of capillary forces to encourage water absorption, or it can enlarge upon water contact, or it can liberate gases directly to disintegrate the tablet. Again, like any other excipients that you add on, the amount of disintegrants that you add into the tablet has to be just nice in a way, because too much of it can cause the tablets to disintegrate in the bottle due to the atmospheric moisture, and too little of it might be insufficient to disintegrate the immediate release tablets. There are two classes of disintegrants. One is the traditional ones like starch, and there's also the newer ones like called super disintegrants such as crosspovidone and etc. The last class of excipients that we're going to discuss on are called lubricants, whereby there are normally hydrophobic materials that are added to enhance the powder flow through the tablet machine by preventing the tablet material to stick to the dye after it's been compressed. Examples of the lubricant family include all from the stearate group, such as magnesium stearate, calcium stearate, and stearic acid. Again, too little of lubricants may not allow the formation of satisfactory tablets because due to the residue of the previous ones that got sticked into it, but too much of it can cause the formation of a hydrophobic coat that can inhibit the tablet from disintegration and dissolution. When you look at the graph, you can see that for salicylic acid, if you add magnesium stearate, it can actually reduce the amount dissolved, and again depends on what you're adding. On the right hand side, whether it's sodium lauryl sulfate or magnesium stearate, again you can cause a different profile of dissolution. I have also added some summary charts on the effects of lubricants, glidants, and anti-adherents, and also on the reason why magnesium stearate, which is the lubricant, is so commonly used, because it has multiple effects on speeding up the manufacturing process and enhancement of therapeutic effects. So, What do you think? Do you think it's possible or easy to formulate a generic drug which has bioequivalence property to the originator? If yes, you can add a +1 at the comment section, or you can write down what you feel about it. Notes for this video can also be downloaded at the link below, and these are the references for the topic that if you're interested, you can go through them as well. Thanks a lot for watching. If you like this video, do share and subscribe to the channel and on the bell. Thank you.